Hey guys! So today we're going to cover Chapter 14. Chapter 14 is a chapter that covers DNA structure and function and how you're able to replicate your DNA to create all of the cells that compose your body. Okay, so last week we covered heredity and how scientists discovered what chromosomes were and what genes were, and how there are multiple genes on your chromosomes and multiple alleles for those genes, and how it can be passed through offspring or through daughter cells. But this week we're going to start at how the genetic information is actually replicated. So in the 1860s, a physician named Frederick Meissner was able to isolate some phosphate-rich chemicals from white blood cells. These were leukocytes, which you probably already know are involved in your immune response whenever you take in a disease or a virus or something. These are some of the first responders, so to speak, in stopping that from spreading throughout your body. We're not talking about that. But, so within these white blood cells, he isolated these chemicals and named them nuclein because they were isolated from the nuclei of the cells. We now know, looking back, that this was actually DNA that he had isolated. Quite a few decades later, in 1928, a bacteriologist named Frederick Griffith conducted a pretty groundbreaking experiment on mice. So he was able to demonstrate, the first time this was ever demonstrated was he was able to demonstrate bacterial transformation, which is a process in which external DNA is taken up by a cell, which changes its morphology and physiology, and then, of course, also changes any daughter cells it may produce as well. So, he was working with a type of bacterium that causes pneumonia. It's called Streptococcus pneumoniae, and he worked with two types of strains. He had a rough strain and a smooth strain, and they were named for their appearance on the colonies, or their growth on the colonies. So the rough strain, labeled as R, was a non-pathogenic strain, and it did not cause this disease. He also had a smooth strain, which was pathogenic, and it did cause the disease. It also had a specialized capsule on the outside of its cell wall that helped it avoid the immune responses of the host mouse. So, with these two strains, first he injected living cells as a control into a mouse. The mouse died. Living S cells, so the pathogenic virus. He injected living S cells into a mouse and it died. Then he injected living R cells into a mouse as a control as well. It's non-pathogenic. It didn't kill the mice. So then he heat killed some S cells and injected those into the mice. Predictably, it did not kill the mouse because they were dead. However, a very, very unexpected result occurred whenever he mixed some heat-killed S cells, so the pathogenic version, with some living R cells. So he mixed them up, put those in the mice, and they died. So what happened? Well, he concluded that there must have been some type of a substance, which he named the transforming substance, that allowed the transfer of something from these dead pathogenic cells into the living non-pathogenic cells, which made them pathogenic and kill the mice. So, of course, now we know that this was actually DNA that was injected into these mice. A little bit later on, in 1944, Avery, McLeod, and McCarthy did another experiment using this transforming substance and discovered that it was indeed DNA. So what they did was they isolated the S strain from the dead mice. They isolated proteins and nucleic acids as well, so that would be RNA and DNA, and they applied enzymes that specifically degraded each component. So it degraded one type of enzyme would degrade the protein, one type of enzyme would degrade the nucleic acid. And then they used each mixture separately to transform the R strain. Now this is what their methodology map, if you want to call it, looked like. So they took heat-treated S cells, and they isolated proteins, DNA, and RNA, and then they used enzymes that would specifically degrade one component out of each of these three mixtures. So over here on the left, they degraded the proteins, and with DNA and RNA, they were still able to get a transformation. So the mice would die, and the DNA of the S strain was able to be passed. So over here in this middle section, they degraded the RNA. So with DNA and proteins, again, they were still able to get a transformation. So the mouse died and the S strain was able to be passed. I guess it'd be backwards. The S strain was passed and then that caused the mouse to die. But then over here on the third section, they degraded the DNA. And so all they were left with was a mixture of RNA and proteins. And whenever they injected that into the mouse, they didn't receive a transformation, so the mice lived. What they concluded from this was that when DNA was degraded, the resulting mixture was no longer able to transform the bacteria, whereas all other combinations were able to transform the bacteria, so the combinations that contain DNA. So this is how they got the conclusion that DNA was the transforming principle from the previous experiment of Griffith. Now, in 1950, a guy named Erwin Chargaff took a look at the content of DNA in several different species of animals, and he was able to draw some overarching conclusions from them. These conclusions became known as Chargaff's rules. So the first rule we're going to look at is that the amount of adenine, thymine, guanine, and cytosine do not always occur in equal quantities. Well, that's because we know now that adenine and thymine bond together and guanine and cytosine bond together, but they're not going to be present in equal quantities. So his next rule was that the relative concentrations of the 4 nucleotide bases will vary from species to species. Why is that? Because they have different genetic makeup. It's going to vary from species to species, or there wouldn't be a difference in species. So there were some exceptions to this, or some rules within the rule, so to speak. So the relative concentrations of these 4 nucleotide bases will not vary within the tissues of the same individual. So the tissues of the individual will have the same relative concentrations of the 4 nucleotide bases, as well as individuals of the same species. They will also contain relative concentrations of the 4 nucleotide bases because they're composing the genetic structure for an entire species. So there's going to be similarities there. The third rule was that the amount of adenine will be equal to the amount of thymine, and the amount of cytosine will be equal to the amount of guanine, what we just talked about. Though there will always be equal amounts of those paired bases, because they're paired. There has to be an opposite for each, or a thymine for each adenine, and there has to be a cytosine for each guanine. So then the fourth rule was that different species will have equal amounts of purines, so the adenine and guanine, and equal amounts of pyrimidines, thymine and cytosine. But they'll have different ratios of the adenine to thymine and the guanine to cytosine. So then in 1952, Hershey and Chase conducted an experiment on bacteriophages. So bacteriophages are basically like viruses that infect bacteria. They are these nifty-looking little robot-looking guys. They'll have a phage head, which is this big round thing, a tail sheath, and a tail fiber, and they'll go sit on the cell or the wall of a cell, and they'll inject their DNA into that cell, and it'll then be uptaken by that cell, and they'll begin replicating bacteriophages. Pretty similar to how a virus works on any other type of cell. So, what they did was take two groups of bacteria. So the first group of bacteria, they infected with a bacteriophage that was radio-labeled with an isotope of sulfur that labeled protein. The second group, they infected with a bacteriophage that was radio-labeled with a phosphorus isotope that labeled DNA. So they took those cultures, centrifuged them, blended them together so that they could separate the phage from the bacteria, and then they cultured those respective bacteria. And what they found was that the culture with bacteria infected with the radio-labeled phosphorus isotope produced radioactive phage, whereas the bacteria infected with the sulfur-labeled isotope did not. So, of course, that meant that DNA was, again, the type of material that was able to pass the genetic information from this bacteriophage into this cell to be replicated. Then, in 1953, Watson, Crick, and Wilkins, along with the help of Rosalind Franklin, discovered the helical structure of DNA. Now, we've already covered a lot of the structure of DNA, so we're not going to go too in-depth over it again, but just as a review, we'll kind of cover it. So DNA is nucleic acid. It's composed of nucleotides. And what you take is a phosphate group, add that to the 5' location of a 5-carbon sugar. The 5-carbon sugar will either be deoxyribose, which is found in DNA, or ribose, which is found in RNA. And then that will be attached to a nitrogenous base. The nitrogenous base will always be attached at the 1' location of the sugar. Then there will also be an open 3' end. And the open 3' end is then used to add another phosphate group onto to build another section of DNA. And of course, DNA is arranged in an anti-parallel fashion, meaning that 5' to 3' will run one direction on one strand, and it'll run the opposite direction on the other. So the two strands are complementary to each other. Now, we're getting into how DNA is replicated. So when they started getting into the nitty-gritty of DNA replication, there were three primary methods of DNA replication that they were considering. So the first would be conservative replication, meaning that the parental DNA remains together with the newly formed daughter strands. So this first graphic over here. Parent strands are together, daughter strands are together. The next method would be semi-conservative replication. In this method, each of the two parental DNA strands will act as a template for the new DNA to be synthesized. Then, after replication, each double-stranded DNA includes one parental unit, or one old unit, and one new or daughter unit. Then, in the last method that was proposed, dispersive, both copies of the DNA have double-stranded segments of parental DNA and newly synthesized DNA interspersed. Now, to answer this question, Meselson and Stahl grew E. coli in a medium. So first they grew it in a medium containing a heavy isotope of nitrogen. After it was allowed to grow for a little while, they then placed it in a light isotope of nitrogen. The cells were then centrifuged at high speeds and loaded into a gradient. A gradient is a solution of salt or sucrose that allows particles to separate based on their density. So the DNA formed into bands according to its buoyant density. So Buoyant density is the density within the gradient in which it floats. After one generation, the single band observed was at an intermediate position between the DNA of the cells grown exclusively in the heavy nitrogen isotope and those in the light nitrogen isotope, which suggested either a semi-conservative or a dispersive mode of replication. After two generations in the lighter isotope of nitrogen, two bands formed. One band was at the intermediate position between the lighter and heavier isotope, and the other corresponded to the band of the lighter isotope DNA. So this could only be explained if the DNA were to replicate in a semi-conservative manner. So let me back up so you can remember what that is. So that means that you have a resulting complementary daughter strand of DNA paired with the old parental DNA. But there's not this intermixing and they're not paired together. So now that we've covered the history of DNA replication and how these experiments were done and these discoveries were made, we're going to get into how DNA is actually replicated. So the replication of DNA begins at special sites called the origin of replication. The origin of replication is composed of a specific sequence of nucleotides. At that sequence, the two strands of DNA will be separated, which will open up a replication bubble. This picture right here shows you an illustration of what a replication bubble looks like. So in eukaryotic cells, the chromosomes may have hundreds or even thousands of origins of replications. So that means that they'll have hundreds or even thousands of replication bubbles. Whereas a prokaryotic cell typically only has one. And for most of this chapter, we're just going to cover how prokaryotic cell DNA is replicated. And then we'll get into a little bit of how eukaryotic cells replicate. But the following few slides is just specifically how prokaryotic cells replicate. Okay, so the origin of replication will be recognized by certain proteins that will bind to the origin of replication site. An enzyme called helicase will then come in and unwind the DNA by breaking down the hydrogen bonds between the nitrogenous base pairs. So this will open up a couple of Y-shaped structures called replication forks. As you can see, both of these illustrations show you a replication fork. This left one shows you kind of where it opens up. So right here would be the origin of replication, right in the middle there. And this red line represents the original parent strand of DNA, whereas the inner blue line represents the new daughter strand. So if we were going to look at these two pictures together, this left one would actually be turned to be in the same orientation that the right one is. So over here you see the parental duplex, parental strands. The replication fork opens up and you will have daughter strands inside. Now there are some particularly important enzymes called DNA polymerases that are essential in the replication of DNA. So these are specific to prokaryotic cells. And DNA pol in prokaryotic cells is an accessory enzyme during DNA replication. DNA pol 2 is also an accessory enzyme for DNA replication, but it's also required for repair, which is its primary purpose. DNA pol 3 is kind of like a pretty big player in DNA synthesis. It is absolutely required for DNA synthesis. However, it has a few restrictions, which will make a little more sense in a minute. So, first restriction is that it must add nucleotides in the 5' to 3' direction. And it also requires a free 3' or a hydroxyl group located at the 3' location on the 5-carbon sugar. So here's another look at what I'm talking about. So here we have the old parental strand of DNA in the blue and a new parental strand of DNA in the green. Down here you have your 5-carbon sugar, and at the 3' location you have an open hydroxyl group. That hydroxyl group is then going to bind to a new nucleotide that DNA polymerase III will add. But DNA polymerase needs a little bit of help to get started. It can't start adding nucleotides to nothing. So what it requires is a primer, so to speak, to be laid in order for it to add new nucleotides to. So this primer is provided by an enzyme called polymerase, and it's just a short chain of nucleotides. After that primer is laid, DNA polymerase III can then start adding new nucleotides to the sequence. So this is an overall view of a replication bubble. A replication bubble is a 2-strand template of DNA that have opposing orientations. One strand is running in the 5' to 3' direction, and the other strand is running in the 3' to 5' direction. But see, there's a little bit of an issue with the replication bubble. Only one strand can be synthesized continuously towards the replication fork. That would be this leading strand, so the longer strand over here. Notice though, it still requires a primer to be added in order for it to be built, but it's a long continuous strand. Now, the other strand that's extending away from the replication fork is a small fragment known as an Okazaki fragment. It's also called a lagging strand, or the strand that is broken up into multiple pieces. It also requires a primer to start. The Okazaki fragment was discovered by a Japanese scientist named Okazaki. That's where it's got its name from. And it actually lays down DNA strands kind of backwards. So the primer will be laid down pointing opposite of the replication fork and of the origin of replication itself. Then the new primers, the new segments will be added in the direction of the origin of replication. Then they'll be added to the leading strand. Now there are some other important proteins at work here. So you have a single-strand binding protein, or a helix destabilizing protein, which prevents the 2 strands of DNA from reconnecting. This happens when the replication fork opens. Then topoisomerases will create nicks in the DNA strands to prevent excess coiling. A sliding clamp protein, which is a ring-shaped protein, will then bind to DNA, and it'll hold the DNA polymerase III in place as it continues to add nucleotides to the growing strand of DNA. Then DNA ligase will come in, and it will seal up the nicks that were created between the newly formed DNA and the previously formed DNA. So, to recap and give you a good overview, DNA will unwind at the origin of replication. The origin of replication is a specific sequence of nucleotides in which a protein will bind to in order to initiate the replication bubble. Helicase will open the DNA, or split the DNA, which will then form multiple replication forks. Single-strand binding proteins will then coat the DNA around the replication fork to prevent it from reconnecting or rewinding. Topoisomerase will bind at the region ahead of the replication fork to prevent supercoiling. Then primase will synthesize RNA primaries complementary to the DNA strand. DNA polymerase III will then come in, and it will add nucleotides to the open hydroxyl group that is on the 3' location of the 5-carbon ring at the end of the primer. Elongation of both the lagging and the leading strands will continue. RNA primers will then be removed by the exonuclease activity. Gaps will be filled in by DNA pol 1 by adding NTPs like ATP, GTP, TTP, or CTP that add energy to drive the polymerization. Then the gap between the two DNA fragments will be sealed by DNA ligase, and you have completed DNA replication. Here is what an actual replication bubble looks like under a microscope. You can see the strand of DNA split, form the replication bubble, and then reunite. Go ahead and take a look at these videos after this current video. Some extra resources for you to hopefully help you understand this stuff. Now we've covered prokaryotic DNA replication, and it is very similar to eukaryotic cell DNA replication with a few differences. So, one big difference is that the origin of replication for prokaryotes is in a single location, whereas the origin of replication for eukaryotic cells can occur in multiple locations. It can occur in hundreds, if not thousands, of locations. Now, another big difference between the two is the rate of replication. So prokaryotes replicate DNA at an extremely fast rate. They replicate 1,000 nucleotides per second, compared with eukaryotes, which can replicate about 50 to 100 nucleotides per second. Another difference is the types of DNA polymerase used. So prokaryotes have a total of 5 DNA polymerase types, whereas eukaryotes have 14 main ones with additional ones as well. They also have a telomerase, which prokaryotic cells do not. Then there's a few other differences down here that I don't expect you to know, but RNA primer removal utilizes different enzymes for prokaryotes and eukaryotes, and they also utilize different enzymes for strand elongation and for the sliding clamp. Okay, now we're not going to completely recover eukaryotic DNA replication. The main thing I want you to know is that they have multiple replication bubbles and multiple origins of replication. So right here you can see there are multiple origins of replication located along this DNA strand, or DNA strands, which open up subsequent multiple replication bubbles. Now, another big difference between prokaryotic cell DNA replication and eukaryotic cell DNA replication is this issue of shrinking DNA. So when a eukaryotic cell DNA is replicated, the typical replication process provides no way to complete the 5' end of the DNA. So repeated rounds of replication produce shorter and shorter DNA molecules. In prokaryotic cells, they don't necessarily have this problem because their DNA is circular, not linear. So it just wraps right back into itself. Now, the way our cells are developed to deal with this issue of shrinking DNA strands is there are these portions of DNA that are tacked along to the ends of it called telomeres. Telomeres are comprised primarily of repeating sequences of nucleotides that code for no particular gene. They're just extra genetic information. So they don't actually prevent the shortening of the DNA molecules, but they do postpone the erosion of the genes near the ends of the DNA molecules. It's been proposed that the shortening of telomeres is connected to aging and death, because eventually, if these strands of DNA are degraded to the point that it is affecting the genes that are there now and no longer just cutting off this extra section of DNA known as telomeres, then it can affect your functional capabilities, because you're now missing portions of a gene that tell your body to do something. And this illustration just shows a better look at what's happening with these telomeres. So up here you start with your parental strands of DNA, replication bubble opens up, you begin synthesizing DNA and replicating it. And here we are at the end, where your 5', because they're complementary to each other, your 5' does not add up with your 3', causing a shortened daughter strand compared with the original parent strand. So these telomeres just act as basically just extra information there that can get cut off without affecting your genes and affecting your cell's productivity. Now, this was not discovered until relatively recently. Telomerase was discovered in 1994 by a woman named Elizabeth Blackburn at UCSF. If you want to get a little more information about this, check out these 2 YouTube videos. And that concludes chapter 14. I hope you guys have a great week. We're getting close to the end of the semester. We're just a couple of weeks away now.